Hi everybody, welcome to our channel Art of Learning Botany, beams the quality and designs the future. I am Dr. Bijaya Kumar Mishra, retired Associate Professor Botany from Department of Higher Education Government of Odisha. Today topic is Sexual Reproduction Flowering Plants, subtopic is Microsporogenesis and uh, development of male gametophyte. Okay, so in this lesson, we'll discuss about the process of microsporogenesis, why it occurs, how it occurs. Then we'll see what is the male gametophyte, where it develops, what is the stages of development, and how the gametes are formed. Okay, so let us first start with the microsporogenesis. In last class, I told you that for the sexual reproduction, a sporogenesis is a fundamental process because it reduces the chromosome number of the male plant, of the main plant, right, the mother plant. So you'll find it is done by the process of meiotic division. So sporogenesis means spore formation. If it occurs in the male sexual unit, it is called microsporogenesis. If it occurs in the female sexual unit, it is called megasporogenesis. So microsporogenesis, which we are concerned today. So microsporogenesis is the development or the process of formation of microspores. So where it is formed? It is formed inside the microsporangia, which is present in the anthers. You know, the anther is the bilobe structure that is attached to the that is attached to the top of the stamen, filament in the stamen, and you can see the structure of the anther. So the anther is a bilobe structure having 4 microsporangia, 2 microsporangia in each lobe, and the anther is encircled by the wall layers. There are 4 wall layers. One is called, the outer one is called epidermis. Next is endothelium. Both are single layered, followed by middle layer, that is 2 or 3 cells thick. Now then is a tapetum. Now this tapetum is encircling the each of these. You see, this is tapetum is more prominent cells. Now each tapetum layer encircles the microsporangium. So the tapetum layer is not continuous. It is continuous around each microsporangium. This tapetum is a very important layer because it is a nutrient layer. You can say that it helps in nutrition. It brings the water and nutrients from the outside and gives to the sporogenous tissue, that is the fertile cells, right? But in addition to that, when tapetum degrades, it produces substances which helps in the maturity of the microspore. That microspore matures to pollen, and the pollen is formed by the secretion, by the degraded product of the tapetum. So it produces sporal proteins and then spore coat materials, and there is another substance called the sporopollenin. So these pollen wall proteins, pollen wall materials and sporopollen, they are deposited around the microspore to make it thick. And sporopollen is very resistant material, and it can resist high temperature and pressure, so it is found in the fossil period. From the fossil periods, the pollen are resistant because they are due to the presence of sporopollen, right? So now what we find, how this microsporogenesis occurs. Now the microsporogenesis occurs from the sporogenous cells. So sporogenous cells are produced during the development of anther. They are formed from the microsporangial initials. Now these microsporangia are diploid. They divide, redivide by mitosis repeatedly till they reach the last stage, that is called the microspore mother cell. So microspore mother cell divides by meiosis. Up to now, its division is mitosis. So the microspore mother cell divides by meiosis inside the microsporangium to form 4 microspores. Now this microspore genesis by microspore formation occurs in 2 ways. That means meiosis occurs in 2 ways, in the sense with respect to the cytokinesis. Right. So there are 2 methods by which cytokinesis occurs. Now the first one is called successive, one after the other. and the third one, the second one is called the you can say simultaneous, together at a time. So what happens actually, the microspore when divides, sorry, the mother spore mother cell when divides, so what is that happens? Now they go to meiosis 1. So two cells are formed by the cytokinesis. So there is a one cytokinesis at the end of meiosis 1. Then these two cells again divide to two separate cells. That means 4 cells. So there is another division in each cell, cytokinesis. So there are two cytokinesis here in the two daughter cells and the first one. So there are 3 cytokinesis occurs. So one is at the end of meiosis 1 resulting in 2. Another at the end of meiosis 2 resulting in 2, 2. So there are 3 cytokinic divisions, cytokinesis. So that is coming to the spore tetrad, sorry, spores arranged in isobilateral manner. I'll show you. So during this development, the sporogenous cells, they become enlarged, they gather callus around them, then they divide. The callus is formed around the spores or spore mother cell. Then these callus also formed around the spores. So callus helps a lot of protection to the spores. Now we'll be seeing that. Now let us see how this division occurs. Okay. Now we'll see the how the division occurs. This is the anther. You see. Next we'll see the structure of anther, process micro. Yes. So this is here. You see. I am going back again. So this is the division. You see here the first there is two cells. The first cytokinesis. Okay. So then there is cell division, sorry, nuclear division. Then this is second cytokinesis, this is second cytokinesis. So at the end of meiosis 2, you'll find 4 cells. At the end of meiosis 1, you'll find the other division. So it is called simultaneous type. Sorry, this is called successive type because one after the other. Now, in the next, in the same slide, you say the same thing here. Already cytokinesis are taking place here and here. This is taking place laterally, so you'll find the 4. This is called isobilateral type of spores arrangement. Now in the next you'll find this type of arrangement occurs in monocots. Okay, this arrangement occurs in monocots. But in most of the dicots there is a tetra type, that is simultaneous type of microsporogenesis. You see this is the single microspore mother cell and this is the meiosis 1. Then there is no cytokinesis till now. These nuclei will divide and at the end of meiosis 2 they will divide by 4. There one cytokinesis and they will form the 4 microspores. So they are arranged in spores. So microspores are arranged in tetrads. This occurs in most of the dicots. So this is called the type of arrangement. One is isobilateral, the other one is called the tetrads. But there are other types also. Now you can see this. Here see other types of arrangement. This is isobilateral type. This is a T type. This is a dicoset type. This is a linear type. So different types of arrangement of microspores are there. Anyway, the microspores are formed by the process of meiosis, and microspores is the first stage of the first gametophytic cell. So this haploid, this is very essential. This leads to the turning in the life cycle of an angiospermic plant which is reproducing sexually. Okay, so we'll find now the important. What I was talking about the callus. What is the importance of callus? So callus is a substance, is a polysaccharide. It is chemically called beta glucan. Now it is deposited and from the beginning of the meiosis and ends at the end of meiosis. The deposition ends, but it continues. Now this callus protects the spores from clumping. Even the microspore mother cell will not clump, the spores will not clump. Particular microspore mother will not clump. Okay. Now this checks from the dehydration. This checks from harmful effect of the hormones coming from the subsequent surrounding sporophytic tissues. And in its absence, the flower becomes, the next generation flower becomes sterile. This is called male sterility develops, right? So this is the importance of callus. Now, up to now, what we have read: microsporogenesis, successive type of cell division, sorry, microsporogenesis, successive type, and simultaneous type, then callus. Okay. Now let us see what are the some questions on this, very quick questions to revise your knowledge now. What you have remembered. So the layer that surrounds the sporogenous cell is called tapetum. The cells from which microspores are formed are called microspore mother cell or microsporocyte. There are dash number of microsporangia in a typical anther, 4, 2 in each. Which is the correct sequence of the wall layers in anther from outside inside? Epidermis, endothelium, middle layers, and tapetum, that is bit C. Then microspore mother cell are otherwise called microsporocyte. Which of the following is not diploid? You see, tapetal cell is diploid, microspore is not diploid. Then microspore mother is diploid, microsporocyte is same as microspore is diploid. So the answer is B. Now callus is chemically known as beta glucans. Then the enzyme that dissolves the callus is called callase, that is secreted by the tapetum. How many times the cytokinesis occurs during the successive method of formation of microspore? 3 times. The first at the end of meiosis 1, next at the time of meiosis, end of the meiosis 2. Okay, so it is 3. Now, let us come to what is gametogenesis and gametophyte development. Right. So, male gametophyte. So, you know the gametophyte already have reached by the microspore. Now, the microspore becomes matured by deposition of these wall proteins, sporopollenin, to form a pollen grain. And the development of the pollen grain is the actually the gametophytic male gametophyte, because the development occurs in the pollen grain. It is full matured now. Now, the development of male gametophyte occurs at 2 stages. One inside the anther, that is inside the microsporangia, one is present, and inside the anther. Then they will be released. Then further development occurs when you reach the stigma of the female plant, right, the carpel. So there are 2 stages, but in most cases the pollen grain divides by mitosis to form a 2 cell stage. So how? There is some arrangement occurs at this time. The protoplasm is reorganized, the nucleus go to the one side, all the organelles, most of the organelles go to one side, there is a vacuole development. Okay. Now, yeah, we will see this one. You say, I'll see this. You see, this is the pollen grain. This pollen grain, there is a thick pollen wall around it, and there are small points which are thin. Now, there is more of the that is what is called the organelles. They accumulate to one side. Now, this pollen nucleus, pollen mitosis occurs here. You can say pollen mitosis to form 2 nuclei, and one small cell is formed. That is called the generative cell, and a big large cell is formed called the vegetative cell. Most of the organelles are present in the vegetative cell. Few are present in the generative cell. Now, in the nucleus of the generative cell forms DNA. So DNA occurs there in the nucleus. Okay, but this vegetative very more protoplasm and develops to the major part of the male gametophyte. So this is 2 cell structure. At some time you will find, after some time, the generative cell is pinched up into the cell of the vegetative cell. So you will find cell within a cell, right? This is a one cell within another cell. So this is cell within the major, that is the generative cell. Generative cell within the vegetative cell. Okay. So this 2 cell stage occurs in 70% of the angiosperms. So male gametophyte that develops inside the anther occurs up to 2 cell stage. That is inside the pollen grain. Okay. The pollen is 2 cell. Then they will be released by the dehiscence of anther. They are borne by pollinating agents to the stigma. But what about the other 30%? Other 30% of the cell or the plants, they have 3-celled male gametophyte. That means the generative cell which is present now in the pollen again divides to form 2 nuclei, that is 2 male gametes or sperm cells. Clear? So that is the 3-cell stage. So that 3-cell stage is found in only 30% of the uh, angiosperms. So at this stage, the pollens are released and they are borne by pollinating to the stigma, right? Now when they reach the stigma, they recognize that it is compatible or not, the interaction. We'll discuss later. So there is a recognition of self or non-self. Then the pollen develops to the full-fledged gametophyte through a pollen tube. If it is two-celled pollen, then the pollen tube germ comes out through the germ pore. The tube nucleus comes to the tip of the pollen tube, and the generative nucleus or generative cell divides by two mitosis to form two nuclei, and two small cells will be formed, and they are called the male gametes or male sperm cells, right? There is a little difference in their size, so they are called the two cells. But if it is a three-celled one, there is no need of further division. Only the tube nucleus will flow to the tip of the pollen tube and followed by the two male gametes, right? Now the pollen tube elongates to this style of the carpel. Okay, so whether it is two-celled or three-celled doesn't matter. The pollen tube develops on the stigma, and they lead towards the male, sorry, female structure, reproductive structure, the ovary. They will lead to that side. So that is the, you can the carpel, where it contains the female spore is present. Anyway, so next stage you will find. So this is called microgametes. So what is microgametogenesis? The formation of microgametes and development microgametophyte or the male gametophyte. This total is called male gametogenesis. Sorry, total is called the microgametogenesis. Development of the male gametophyte and formation of microgametes. Now you see this diagram. You'll see here there are two sperm cells. The tube nucleus is not clearly visible. Now there is another term coming, the male germ unit. This term was coined by Dumas in 1985 and with his workers. Now this is called the functional unit of fertilization. So this is called functional unit of fertilization is called MGU, that is male germ unit. So what happens? These two male gametes and the tube nucleus, they make a block, they move together in group. Now this bigger one is called the SVN, that is sperm cell associated with the vegetative nucleus, that is bigger, and the small one is called the SWA. The first one, SVN, fertilizes or unites with the polar nucleus, whereas the SWA fertilizes the egg, the male gamete, sorry, female gamete, right? And this complex, due to this complex formation, their movement is coordinated and fertilization is coordinated. The fertilization and movement is coordinated and they directed the fertilization process. So this makes a coordination, and there is a preferential fusion of the small one with the egg. So that is called the male, that is significance of the male germ unit. Clear? So by now we have read what is male gametophyte, that is the first male gametophyte is the pollen grain. It is two-celled or maybe three-celled, then transferred and further development occurs in the stigma. Then we know how the gametes are formed. Then what is germ, male germ unit. Clear? Now we will see some questions. That is, let me zoom in so that you can clearly read. Now, yes, this give one word. So which is the first male gametophyte in the flowering plant? That is the gametophyte is the pollen grain, not the microspore. Microspore is the fat gametophyte cell, not the gametophyte. How many sperm cells are formed in male gametophyte? 2. Which is most resistant hard substance found on the pollen wall? Sporopollenin. What is the larger cell of the male gametophyte called? Vegetative cell. Which cell of the male gametophyte produces the sperm cell? The generative cell. From which cell the gametophyte pollen tube comes out? Vegetative cell or tube cell, you can say. Now, who used the term MGU? Dumas. Which of the sperm cell fertilizes the egg? SWA, the small one. Which of the sperm cell fuses with the polar nuclei, SBN, the bigger one? Which is called the functional unit of fertilization, MGU, male germ unit? What is the position of the tube nucleus in a germinated pollen grain on the stigma? The tip, at the tip of the pollen tube. Okay. Then find the odd man out in the following set: tube cell, microspore mother cell, generative cell, and microspore. So microspore mother cell is odd man because two and other side haploid, but this is diploid. Now the other question is 1 to 2 line answer. So define microsporogenesis. This is the process of formation of microspores occurring inside the microsporangium, inside the anther. What is the microgametogenesis? Formation of male gametes and development of the male gametophyte. Now what is the vegetative cell? It is the larger cell present in the male gametophyte that is inside the pollen grain. What is the generative cell? It is small cell present inside the pollen grain that gives rise to the male gametes, two male gametes. What is callus? Callus is a chemical substance called beta glucan, which is present on the wall of the spore mother cell, microspore mother cell. Okay. What do you mean by successive type of microsporogenesis? The microsporogenesis where the cytokinesis occurs at end of meiosis 1 and at end of meiosis 2, at both. Okay. So, and it leads to formation of isobilateral type of microspores. Name the different wall layers of the anther. From the outside is epidermis, endothelium, middle layer, and tapetum. What do you mean by MGU? It is called male germ unit, the functional unit of fertilization. Write any two significance of callus. Callus helps in protecting from clumping of the microspore mother cells and protects from dehydration. You can write protects from harmful effect of hormones, and also it checks male sterility development. Now, what is two important layers, two important functions of tapetal layer? It forms the produces a degradative product of tapetalum, helps in formation of pollen wall materials and sporopollenin, and produces a callus, that is enzyme to degrade the callus. Right. Now, why sporogenesis is essential in sexual reproduction process in flowering plants? Because it reduces the chromosome number. Unless it is reduced as the chromosome, there will be polyploids. So it has to reduce it to maintain the balance of the chromosome in the throughout the species. Now, why the pollen is called the gametophyte whereas the microspore is not? Because microspore does not produce the gametes. The gametophyte pollen produces the gametes during further development. So therefore, which produces the gamete is called gametophyte. So microspore does not produce the gametophyte, right? So pollen is called gametophyte because in development it produces the gametes. What is the importance of callus in microsporogenesis? Already I have told. Name the components of a three-celled male gametophyte in flowering plants. Three-celled, one is vegetative cell, big one, and the two smalls called the male sperm cells. Now, give one distinct point to differentiate the successive and simultaneous wall formation during microsporogenesis. The place of cytokinesis. In successive type, there are three cytokinesis, one first at the end of meiosis 1 and two at the end of meiosis 2. And in simultaneous type, there is at the end of meiosis 2. So that is the main difference. Here you can add in simultaneous type, you'll find spore tetrads, but in successive type, you find spores are arranged in isobilateral manner. Okay. So we finish the questions. Now, up to now, we have read the microsporogenesis and male gametophyte development and microgametogenesis. We have seen the importance of callus and we have discussed the different types of questions also. So if you like, subscribe it, tell your friends and subscribe and share it. And I hope you will join for the video session 2. We will be posting regularly the video sessions in Monday and Thursday. Okay. So I hope you will like this video again and give your comments also. Thank you. See you in the next video session.